So g'day guys, my name's Justin Tierney, and today I'll be presenting my master's thesis titled The Influence of Magnesium on the Expression of Growth-Related Genes in Sibling Meagre with Varying Growth Performance. Now, there's a lot going on in this title, and I'm happy to break it down, but first I want to dive into the problem. So what is the problem is varying growth performance. So Meagre especially are a faster growing fish, and in aquaculture they experience this large growth variability. In the environment, in the wild, this is pretty normal and actually desirable for maturation of that fish to later into this sexual maturity. However, in aquaculture, it's generally undesirable as the subordinate fish tend to be picked on, and they take a lot more time and resources to get to a bigger size. So how do we solve this problem? Size, yeah. So we've turned to genetic features to try to understand and gain a better idea towards the growth potentiality, and even try to see whether or not these fish have this sort of genetic predisposition to growth. So exactly how do we do that? Well, we can refer right back to the central dogma theory of biology, which essentially, really briefly, states that DNA is transcribed into RNA and then translated into protein. There are epigenetic functions that impede transcription and translation, and I won't delve into those too much. They're very interesting for future research. However, we mainly focus on the RNA aspect of this dogma. So we have sibling Meagre with varying growth performance, and we try to use the expression of particular genes to quantify that variation. In the study, we also have the added influence of magnesium. So we have three null hypotheses. The first is that there is no effect of growth rate on gene expression. The second is that there is no effect of magnesium on gene expression. And the third is that there is no interaction effect between these two on the genetic expression. So in this particular study that was conducted in 2018, fish were part of a 12-week growth trial where one group was exposed to a 1% inclusion level of magnesium, whereas the other group was not. After the 12 weeks, they found that there was this large difference between this large varying growth, and inexplicable to the added magnesium. What we decided to do was separate these growths into certain classes, and we can see here that the only significant difference between the groups was observed in the medium growing group. Whilst these trends are still obvious in the slow and the fast, they're not significant. However, it's important to note now that we only took samples from the slow and the fast growing groups for analysis. We did try with the medium, we were unsuccessful, but so 12 fish from each of the slow growing magnesium, slow growing control, slow growing fast magnesium, and the fast control, for a total of 48 fish. So, liver and intestines from these two, and these are the genes that we looked at: insulin-like growth factor 1, IGF binding protein 2, somatolactin, prolactin, and as a reference gene, GADPH. So probably thinking, how on earth do we even try to identify these genes, or how do we even know which genes to look at? Well, to understand the HPS axis, or the hypothalamic pituitary somatotropic axis, which is important and regulates growth in all animals with an endocrine system, even humans, and especially in fish. So essentially what happens is an environmental stimulus is picked up by the hypothalamus in the brain, which is then sends a signal to the pituitary gland, and in this case producing growth hormone. Now, growth hormone has two potential paths it can follow. It can either go directly to the skeletal muscle, or the most common direction is straight to the liver. Now, because we had liver samples, it was important to understand that, important to realize that the liver has the receptors for the growth hormone, but we are looking at what it can produce. So we look at the IGF-1 and the IGF-BP2, which is produced as soon as growth hormone is attached. These molecules then travel around and induce growth in the skeletal muscle and the dissolution of fat tissue in help using this fuel as growth. I will also mention here that there are specific regulatory genes for each of those processes. They're called sirtuin genes, and I won't get into too much, but obviously are important for future directions. So we had the liver, we also had the intestine samples. Now the intestine is very important for osmoregulation in fish, and so prolactin is the major hormone secreted in the intestine that helps with the absorption of minerals and ions. And because we're looking at a magnesium study, we've got this added effect of magnesium. I thought it was very interesting to maybe look at the expression of prolactin in the intestine to see if that influenced the growth in fish. There's also the added looking at the hormone somatolactin. Now, somatolactin is fairly new, so skeletal muscle. So we thought we'd look at it in the intestine just to be safe. So we have the what, the where, the why. Now we understand the how. And this is essentially we look for these genes by identifying primers. So we design primers on known coding domain sequences found in NCBI. We can then use NCBI's primer blasts to create primers. We also had to manually create some primers using the criteria you can see under amplicon selection. And then we can check for the efficiency in Vector NCBI, which is a software that looks at binding efficiencies, secondary structures, and then we can also run primer efficiency tests. So we have the RT-qPCR pipeline next. RT-qPCR stands for the reverse transcriptase quantitative polymerase chain reaction. And it's different to normal PCR because we have this reverse transcription process involved as well. And this essentially means that we need to reverse transcribe our messenger RNA that we extract. If we can remember back to the central dogma, the RNA is the intermediary between DNA and protein. We look at trying to quantify that expression. So mRNA is extracted from liver and the intestine. It's purified to get rid of any genomic DNA that might be around. Then we quantify its potential contamination and its concentration of RNA. And the most important step in this RT-qPCR pipeline is the cDNA synthesis. So you can see in the bottom right here, we have a strand of mRNA, and we want to reverse transcribe it to complement it. We then separate this, and the original RNA is washed away, allowing us to create a cDNA strand, which is used for the qPCR process. So we have our cDNA, we have the primers that we've designed. Now we need to quantify them in a PCR machine. And this is done by adding all of these with enzymes and buffers into a sample well. And then we produce a CT value. Now, a CT value stands for cycle threshold. And it's essentially the quantification of how much of that gene is being amplified. And when it reaches a certain amplification level or this threshold level, it records the amount of cycles that it took to get to that level, and thus giving us the CT value. So a sample with with a higher CT value actually has less original RNA in the sample. So once we to begin analyzing this data, we look at doing the delta-delta CT method, which takes into consideration the reference gene. This data also went another additional standardization step, mainly because of the high amount of biological replicates. And then we analyze this using a two-way ANOVA and differences in a post-hoc Tukey's test. Now, there's a lot going on in the results, especially because we have control magnesium, slow and fast-growing fish, 4 different genes, and then we're looking at it in liver and the intestine. But I just wanted to mainly point out the highlights of what we found, and that is that if we can remember back to our first null hypothesis that there was no effect of growth rate on gene expression, we can nullify that by looking at, just for 1 second ignoring the magnesium, and looking at the control group for the liver on the left, and comparing the IGF-1 expression from the fast-growing group to the slow-growing group, and we can see that there is a significant difference there. The other genes, looking at the control and at the control group for the other genes, we can't notice much of a difference between slow and fast growers. And to move on to our next hypothesis of our next null hypothesis that magnesium doesn't have an effect on genetic gene expression, we also can't look towards the liver, mainly because there aren't significant differences there. So I prefer actually looking at the intestinal data because we have a little bit more of a more detailed picture. And the first thing I want to note is that the y-axis is an order of magnitude different to the liver expression. However, it still paints a pretty good picture. So the main thing I wanted to point out here was this sort of uniform expression. Obviously, there are still significant differences between somatolactin and prolactin expression for the control group. Now, if we compare this sort of expression ratio, rather, to the magnesium group, we can see that the IGF-1 for the fast-growing magnesium group is much more highly expressed. And it's interesting because prolactin and somatolactin are also repressed. And we can see here that IGF-1 is almost stepping in and acting in a compensatory manner to help with the growth, especially because the magnesium being reducing the expression of prolactin and somatolactin like we hypothesized. So our main findings are that IGF-1 is a good bioindicator for growth, and somatolactin, prolactin, IGF-1 are good indicators for slow and fast-growing fish under the effect of anti-nutritional factors in the intestine. So there is a very big difference between what we can look at in the liver and what we can look at in the intestine. And thus we can conclude that our interaction effect between magnesium and growth rate on gene expression is significant. So the future directions of this study would like to be focused more towards looking at the expression of these growth potentials in a non-lethal way. We have all the samples that we had to look at required the death of the animal. So by using our work, we can try to understand which circulating hormones are probably going to be good indicators for growth. This can also we can also look towards the sirtuin genes that I mentioned before, and the epigenetic functions that regulate those sirtuin genes that can control growth. Personally, and this is probably almost geared towards you, Jeppe, that I would like to see this data being used, or these techniques being used more in fisheries ecology, and especially molecular fisheries ecology, because I feel like it gives us a very nice understanding into the life history or life traits of a particular target species, and can also provide us with a real-time analysis of health or growth potentiality of wild fish. I would just like to say a massive thank you to everyone involved in the project, especially Katia Marques. She has provided overwhelming support, especially throughout a pandemic. And Katarina Marques, these two women were absolutely great to work by and provided so much support throughout the entire thesis. Dr. Marisa Barata for providing the original data, and everyone at IPMA staff that was involved in the study. Thank you very much.